All right. Welcome, everyone. Getting ready a little bit late here today, but I think we're good. We had like 6 people in my house today, not on purpose. There was a breakdown of both of our cars. We left lights on in both of them, and and then the cleaning company sent 2 maids. And then there's the babysitter, and then there's us. And I was like, oh man, are we getting close to too large of a gathering? We weren't really gathered. I mean, everyone was in separate rooms, but it's a lot. All right. Well, not sure if there's anyone here. I'm just talking to. Oh, apparently this doesn't know if there's anyone here either. Okay, good. There are people here. I can see you. Okay, great. It was like cannot load. All right, great. Well, Angie, real quick, I did look at that and give everyone credit for that very confusing homework question. Thank you for bringing it to my attention. I didn't realize it was giving wrong answers. It definitely is wrong. There's some very confusing pieces. So we'll walk through the homework today, but I'll explain what we should have seen. Okay. Let's move forward into, we already started gene regulation, but we can continue that. And then we're going to march into the last one that we're going to be working on, which is genetic engineering, basically. Genetic recombination and genetically modified organisms and CRISPR-Cas9, which will form something interesting next week. Okay. All right. We talked about these various things. All right. So eukaryotes have extra regulation layers as part of their having chromatin and chromosomes, separating transcription, translation, processing mRNA, and many more post-translational modifications to proteins available in eukaryotes. So we're going to talk a little bit about some of that. This is not going to be as in detail as the next one will be. There's a lot to go through for genetic modification and gene recombinant DNA technology. So we're going to talk through all of that most of the rest of the class. Okay. All right. So in chromosomes, there are these discrete domains, which is occupied by each chromosome in the interphase nucleus. Okay, so each chromosome has kind of a spot that it hangs out, staying separate from other chromosomes. And these interchromatin compartments, these are channels between chromosomes. And so the idea is that the transcriptionally active genes are located at the edge of the chromosome territory next to channels. And that facilitates coordinated expression across multiple chromosomes. So the mRNA and RNA polymerase can travel around 2 different chromosomes immediately, kind of like little mRNA like express trains, okay? So it facilitates coordinated expression of multiple genes on separate chromosomes. So this allows for, in a more complex genome, these things to happen, okay? All right. So transcripts produced at the edge of the chromosome then move into the interchromatin compartments. And the interchromatin compartments processes everything right away. And so that does the capping and splicing and poly-A tailing of multiple RNAs. And then that is shipped off to be read by the ribosomes. Okay, so this is sort of a coordinated way of doing things, a bit more organization than a bacteria would have. All right. Chromatin can be altered in order to make it more accessible. We talked a bit about this. I'm going to revisit those, but the idea is that closed chromatin has a tight association of DNA with histones, okay, that affects transcription. Opening the chromatin is important to initiating transcription. So you can often switch between the open and closed due to this histone composition. And again, the facultative heterochromatin does this all the time. Consider the facultative heterochromatin as sort of the ones that we have to induce to be ready through changes in chromatin because we don't use these as often. Okay. Changing the histone composition of nucleosomes can influence transcription. Basically, RNA polymerase is going to attack any of the DNA wherever it can and start transcribing, so you have to keep it from doing that if you want to have specific gene expression, specific not. In order to modify the histones, you covalently bond these functional groups onto the tails. We talked a little bit about this before. Acetylation is generally going to increase transcription because it opens it up. Methylation is generally going to decrease transcription because it closes it off. Okay, so most common is acetylation, methylation, and adding phosphates. Okay, so decreasing the positive charge with acetylation reduces the affinity to the DNA, and then methylating it increases that positive charge, increasing the affinity generally, and then doing the phosphate one just makes it more reactive overall. So these are the things these are the restructurers of chromatin. Okay, these are called HATs, histone acetyltransferase enzymes. And these enzymes will add the acetyl groups onto the histones, opening up that DNA section, okay? So these are associated with increased transcription because they are acetylizing the histones. The histone deacetylases takes off the acetyl groups and can encourage decreased transcriptions. So the idea here is that when they remove it, this reduces transcription of the genes that are associated with them. Remember that even though we don't have the exact same setup of those operons as bacteria, we do still have these little proteins that are transcription repressors. Those transcription repressors, there are some places that they can block the promoters, but we don't have operators in the same way that bacteria do. Instead, some of the transcription repressors can recruit deacetylases. Some of the promoters, the ones that activate the gene, can actually get the histone acetyltransferase enzyme. So this is one way that we can turn genes on and off in particular. So you can also do chromatin remodeling. So this is the previous one was the opening and closing of histones, but the remodeling involves repositioning or removal of nucleosomes, okay, those little things that the DNA are wrapped around. And so that can open it up and make it more accessible. Think of the polytene chromosomes we saw before. Those have almost no nucleosomes or histones on those, making it so that they're not wrapped tightly. So again, transcription regulatory proteins can access these, turning on and off certain segments. Again, also that transcription regulation. You have the activators and the repressors that can attach then and reduce or increase transcription of a gene. So you can reposition those to regulate it instead of at the base level of chromatin. You can reposition the chromatin now to have the different layer of actually repressing on the DNA itself. Okay. All right. So here we have the acetylation. Okay. More acetylation means the histone tails are off. And you have this activator that also acts to move apart these histones, opening up the DNA. And with more methylation, you still have some availability of access, but it has been reduced. So we have active. Permissive can be transcribed, but is not transcribed as much. Repressed, very little acetylation, more methylation, and then the inactive is phosphorylated to basically make these histones now group together, and now you have a very tightly coiled and tightly bunched segment of chromatin. So this is the whole process of chromatin remodeling. All right. So we talked about promoters a little bit on Tuesday because we were discussing that activity that we were discussing the case study. But these promoter elements, again, this is a region of DNA that's recognized by these RNA polymerase II transcription factors. Okay, remember the different polymerases. Those will create mRNAs for various things, depending. But this is required for transcription initiation. Promoters are always immediately adjacent to the regulatory gene. So if something has an error on the beginning of the gene, then that could be affecting the promoter, affecting the extra production of the protein. All right. So you've got both the core promoter which helps to initiate the transcription, but also you have the proximal promoter elements, those binding sites for sequence-specific DNA binding proteins, and that has to do with all the activators and other pieces, okay? And then there's also a way that, again, an interesting piece is this is sort of a different way of alternative splicing, if you will. The idea is that you have focus promoters specify one specific transcription initiation site. Dispersed promoters specify weak transcription initiation at multiple start sites. This can create multiple transcripts. Know that they all contain the gene. Just the promoter length is different. This can actually mean that this will promote multiple creation of mRNA to increase the gene products. And the separate activator of these dispersed promoters will promote that as well. So promoter mutations strongly impact transcription. Remember, we talked about this as part of cystic fibrosis, is that the levels of transcription, if there is anything related to the promoters, either upstream of the Tata box, the specific promoters, or any of these boxes that would be identified by specific RNA polymerases, all of that affects transcription rates heavily because if it can't be identified as a site for RNA polymerase to attach, then you have decreased transcription. So promoters are extremely important, and that's why it was why the person in our case study had such issue. She realized that actually that the promoter mutation that she had was going to create it so that she had very low expression of the gene, because she had the whole gene there, but not the promoter proper. And that means very low transcription, and that means that cystic fibrosis would express. All right. Last thing I want to talk about is a little bit of control on errors, okay? So I talked about how mRNA doesn't stick around very long, but eukaryotic mRNA does stick around a little bit longer than the than bacterial. Okay, so bacterial is very rapidly degraded, but regulating how long the mRNA lasts in the cytoplasm is important. The idea is that if you want it to be expressed a lot, actually putting a longer poly A tail on it means that it'll degrade slower and it'll be read multiple times by the ribosome. More robust means more robust mRNA. So overall, it lasts longer. And so this is, again, regulated through the poly-A tail addition. You add a longer poly-A tail, and it has more time before it breaks apart. This allows for it to be read over and over, and so if you, basically there are signals for when you need high expression or moderate expression of a specific gene, and adding a longer poly-A tail will make it last longer in the cytoplasm, so this will mean higher gene expression through fewer mRNAs. And so there's something called deadenylase. It takes off the polyadenal tail, basically. So the presence of the deadenylase is important. You don't want random mRNAs that have been there forever floating around. The idea is that if you have a longer poly-A tail, then this deadenylase takes a little bit longer to break it down, so you can have multiple translations from one. But once it's broken it down, there is ribonucleic decay, and also a decapping enzyme, and eventually it's broken apart for parts. Your body recycles everything, puts all the pieces together, and ships it back to be made into new mRNA. Okay. But this is important to actually have this, because without having a specific way to destroy it, then if you only needed, let's say, the breastfeeding protein for a couple of months, you can accidentally be producing breast milk forever and ever, and that's no good. So you want to be able to turn it off. Our body has these setups in order to control and reduce the amount of excess proteins created after it's not needed. Okay, that's the turning off of the mRNA, not the turning off of the gene. Turning off the gene is separate. All right. With these, an interesting piece is that sometimes mRNA doesn't transcribe properly. It's not based on the gene, but occasionally RNA polymerase makes a mistake. It cuts off too early. The mRNA is degraded before it is capped and tailed. And so you can have these problems, and also sometimes you can have nonsense that comes from the RNA polymerase itself. Okay. So again, errors in transcription can either be caused by mutations or by RNA polymerase. This can occur in everyday cell activity. Again, remember that mutations can happen in your specific cell DNA outside of the transcription, outside of having a mutation in your genome, okay? So in order to reduce the impact of these accidents in single cells here and there, your body has a way of detecting when something bad is going on, okay? Creating erroneous and truncated proteins are wasteful. If there's a nonsense mutation in particular, it can chop off the protein early, and it's useless for your body to be creating these tiny proteins that are eventually just going to be degraded because they don't work. Or worse, that these proteins can actually be malformed and lead to cancer. So a way of your body controlling a cell-specific error is to do mRNA surveillance. So we have a defense against this, okay? When translation terminates very far from the poly-A tail beginning, or upstream of a targeted exon junction, the mRNA will be tagged. And this means that rapid exoribonuclease degradation is induced. Basically destroy it on site. It's sort of think of it as an antibody, but for specifically broken mRNA. This prevents further translation of the mRNA, turning it off quicker than, say, the poly-A tail degradation would. This can eliminate aberrant mRNAs, and also it's a faster switch to turn off gene expression if you want to just destroy the mRNA because you have a signal that you don't need it anymore. You want to destroy all those that are tagged. then you can just turn it off immediately instead of waiting for those to degrade. All right. This is a nice one here again. So we have the mRNA here. Okay. These exon junctions. The exon junctions, again, these are little tiny proteins that have been created that they highlight and they say, okay, here's the start of an exon, here's the start of an exon, and here's the stop, okay? With the normal transcript, as the ribosome travels down, it displaces these exon junctions. Great. You have a proper production of the protein. But if this error here makes it stop early, this sends out a signal and says, hey, I wasn't knocked off. Nothing knocked me off. And so if it's on there too long, it starts to attract these other markers. And these markers will then tell the ribosome and other exonucleosomes to break it apart. Say, stop, actually this is marked to be destroyed. So that's actually very helpful. All right, and then we already discussed other options. We talked a lot about alternative splicing, talked a lot about activators and repressors, and we haven't focused this class on post-transcriptional modifications, but gene products can be edited that way. Okay, so the idea is that after you translate it, you can refold and add on a few extra things to the proteins themselves. All right, so that is all of gene regulation. Mostly it's just focusing on the ways that our body is able to fix errors. Think of our body as sort of like a car that, I don't know if you guys have any of the newer cars. Sometimes the car will send you a text message if it's experiencing a problem. I had a car like that once. So it would send you a text message and say, hey, bring me in for maintenance, at low oil, or there's an error in the engine, or something like that. And I was able to take it in with information from that. So your cells are constantly communicating with each other and communicating within the cell to say what it needs. And in some of this gene expression part tells you why certain people will have various gene expressions, and also tells you a little bit about why even these gene mutations happen. All right. Does anyone have questions about the gene regulation overall? Again, some of your homework will have to do with the targeted mRNA destruction and discussing the different ways that the lac operon works and other pieces like that. Okay. There's also a little bit about DNA, a little bit about recombinant DNA. Okay. So that's next week's homework. Next week's homework is technically extra credit. I recommend you do it. Additionally, I was looking through numbers and I realized I was supposed to post a review quiz this week, and I didn't. But it's okay. If the review quiz will be, I'll post a review quiz next week, and instead of having a review quiz for, we'll have the review quiz include gene regulation and include gene mutation. Post-transcriptional modification is not the same technique because CRISPR-Cas9 actually works on DNA and not on mRNA. And the post-transcriptional is after translation to a protein. That's when your body can actually edit proteins after they are already created without using a mRNA transcript. CRISPR-Cas9 we're going to go through. We're not getting to it today. There's a lot to go through before we get to CRISPR. What we will go through today is kind of the introduction to some of the pieces that have to do with CRISPR, in particular some of the exonucleases and the restriction enzymes. Okay, So CRISPR-Cas9 uses some of the tools that are used in other parts. And then we're going to discuss CRISPR-Cas9 on Tuesday and also a CRISPR-Cas9 case study. Again, the case study is going to replace, I guess technically I was only supposed to have 9 case studies because we had the previous mitosis meiosis. So the CRISPR-Cas9 case study is replacing one of the quizzes, basically. The Blackboard quizzes. So you'll have one Blackboard quiz that's just going to cover a broader range of stuff. Gene mutations and gene regulation and so it's going to cover like 3 chapters. Okay. Let's start moving on here. I haven't posted the next one yet. I was editing it because I was finding so many interesting things, and so it took me a little while to do things that I was happier with. So these are some of the pictures. Oop, that's not how I wanted to set it. Hold on. I just forget to set up the slideshow before I pull these up. Here we go. Okay, set. All right, so we're going to talk about molecular genetic analysis, biotech overall. Okay, so hopefully maybe this will get to some of your questions about CRISPR. Again, there's just 2 more full lectures left. I'm hoping I'm going to get through everything that we're interested in by next week, but we might not. So as far as this, I am going to be creating a small handout to complete for in terms of restriction enzymes, which is one of the first things we're learning about. Some practice questions that'll be our class activity. And so that was just going to be a worksheet. It's not going to be a complex, like here, write this paragraph sort of thing. Okay, it's just going to be a short worksheet that you'll get 5 points for, because I think I have it scheduled for a total of 1 this week and 1 next week for activities that you can do. Okay. Let's make sure this is as big as we can get it. All right. So techniques of modern molecular genetics have revolutionized biology. We've already talked a lot about it in our case studies as it has become more and more relevant with some of the pieces that we're talking about. Recombinant DNA technology, remember that's what created insulin. So these are genetic materials from 2 different sources, often 2 different species, not the case for insulin. That was the case of more making the bacteria build it for us. But often 2 different sources are combined in the same DNA molecule. This would be sort of the, I guess, human DNA to bacterial DNA. But we can use bacterial DNA also to fuse things into other organisms. So techniques directly manipulate the genetic material to locate, isolate, alter, and study DNA fragments. One of the interesting ones that's used a lot that you guys actually used in Bio 1140, if you took it here on campus, is this green fluorescent protein, GFP. You'll recognize it from the GMO experiment that you did to make the bacteria glow. That was the one where you shocked the bacteria, had them pull up plasmids, and then saw how many of them actually were expressing now the green fluorescent protein by putting them under a black light. Now, while we did it only with other bacteria, taking that green fluorescent protein from jellyfish and putting it into just bacteria to make them glow, we can actually use the bacteria that have those to then transfer the plasmid into other organisms. Okay, so that would be the creation of a GMO. GMO being an organism and not just a genetically modified recombinant bacteria. Now we're using recombinant bacteria to make organisms that have this new chunk. Okay? So an organism whose genome has been altered by the transfer of a gene or genes from another organism is called transgenic. I mention this because this is a form of basically human mitigated horizontal gene transfer. We talked about horizontal gene transfer in class. That this horizontal gene transfer being where this, this slug that got infected by the same bacteria that its algal food had was able to get algal genes from it. This happens in the wild in general. This is a way of directing that same process to occur under coordination and in the laboratory. Okay. So biotechnology, again, it harnesses the power of this molecular genetics, all the pieces that we have been discussing about how organisms fight off and also change around their genetics, can be used to manipulate gene products to be created by bacteria, or also to transfer genes from one organism to another. This can create useful products, okay? So we can all understand the process of inheritance and gene expression by doing this. We've been talking a lot about the treatment of disease and correcting genetic disorders using molecular genetic processes, the gene therapies that we discussed. And forensics as well. We are going to be talking some about how DNA testing is done to target and sequence and highlight different tags on your actual genetics. And it can provide economic benefits. Who can think of an economic, so I mentioned plants and animals, but what kind of economic benefits could come from genetically modified organisms? Think about the GMOs that you might eat every day. What's the benefit of those? More food. How more food? Who knows what kind of GMOs you eat on a regular basis? What kind of GMOs? We're not eating actually the strawberries, that's a polyploid. Bread has, it depends on the kind of bread. Actually, generally wheat is not transgenic. Corn is transgenic though. Strawberries themselves, actually, creating a polyploid plant like I talked about, those giant strawberries you find in the store being octoploid, that's technically not genetic transgenics because it's just a copying of the strawberry. We're just inducing the strawberry to become polyploid. That's not actually genetic transgenic technology. Some rice. We're going to talk about golden rice. Some rices, generally not the rice that you're eating in the store. Corn, soy, cotton, and tobacco are, let's see, oh wait, canola. The plant that creates canola. Those are the ones that are the main ones that are transgenic and are on the market today. But the wheat that you eat, strawberries, rice, not. Okay. Dairy products. It's an interesting one. A lot of cows are fed GMO corn. They are not themselves transgenic cows. Instead, they eat GMO corn. It's a little bit different. Okay. We are not transgenic ourselves because we consume GMO products. The one you may be thinking of is the growth hormone. Growth hormones, while created through transgenic technology, are not themselves making the cows transgenic. Okay, you'd have to manipulate the cow DNA to do that. Okay, so genes for pest resistance can be inserted into plants. There was so there's definitely a salmon that was created, a farmed salmon that was created as a GMO product. It's not on the market, but they tried to get it to market, but it was rejected by consumers. It's one of the things is that sometimes people just don't want to buy what you're selling, right? But yes, GMO salmon had been created. I forget what the GMO salmon were even supposed to do. I could look it up. So then we have a gene used to alter bacteria for cleaning up toxic waste. This is a good one. If you guys are old enough to remember the BP oil spill, you were younger, but the BP oil spill was a big deal. It dumped tons and tons and tons of oil into the Gulf, and and it was very difficult to clean up. So one of the ways that they were able to start cleaning up certain areas was actually introducing a bacteria, a transgenic bacteria that could metabolize oil, and it would eat oil for food, and it was a way of cleaning up the ocean. Another one is to actually create a protein. Remember, this one's going to be a little bit closer to, these next two are going to be a little bit closer to more like the insulin, using bacteria as factors to create specific proteins, okay? So creating a protein that dissolves blood clots in heart attack therapy. This one is more using bacteria to be protein factories. Isolating an already existent protein in humans and amplifying it to be used as a medication, similar with the human growth hormone and insulin. All right, as we talk about this, I'm going to be giving you as many facts as I can. There's a lot of sensationalism in the media about this. If you type biotech or GMOs or CRISPR into your search box and look for recent stuff, you'll find this kind of stuff. Would I want to designer baby? CRISPR, gene editing, and you. Designer babies, gene editing, and the controversial use of CRISPR. Hidden health dangers. A former ag biotech insider wants his GMO crops pulled. Gene editing is here. It's an enormous threat. This is attached to so this is from the Washington Post. It's an opinion article note. But these sensationalist articles like, gene editing, so scary. And they highlight now, I'm not going to defend this guy, but they highlight the scientist that has, he supposedly did gene editing on twin babies to remove a susceptibility to HIV. Now, this is unethical in the sense that human testing and creation of humans that have been genetically edited is not yet considered safe because there has been not enough information and testing and talking and working to make sure that this is not dangerous or other pieces. Okay, there's a lot to do about humans. So highlighting this scientist, yeah, he did something extremely unethical and against the code and all sorts of stuff. But here's the deal. Not every scientist who's working with this is doing that. But it's easy to point out the individual flaws. You speak about GMOs and people will tell you about Monsanto and how they're an evil corporation. Well, Monsanto's not the only people that are making GMOs, and a lot of scientists are working to get GMO technology actually into the hands of farmers and out of the hands of big corporations. So it's a tool. It's not just a sensational, we're creating designer babies. Okay, we're going to watch a video on Tuesday in relationship to the case study where the person who actually created CRISPR-Cas9 talks about possibilities in how it can be used to better health and help with genetic disorders. And at the very end, she mentions that enhancements are a possibility, but very far away. But most of the media surrounding it centers on the enhancement designer babies concepts instead of the disease regulation and mitigation things that she focused on. Again, the real story is that a lot of the people who actually created the biotech had disease treatment, food production improvement, and human health in mind. Recognize that these are tools, okay? Doomsday scenarios, scientists creating designer babies, these are extremes. They are easily politicized, heavily scrutinized, and focused on by the media and in pop culture. It's kind of how my husband's a physicist, and every time there's a new thing talking about neutrinos or some other buzzword there, they're like, is the universe a hologram? Can we do time travel? But in reality, most physicists are not trying to figure out those pieces. Instead, they're putting together, mysteries of the universe and trying to understand how these pieces work and, creating quantum computers and all sorts of things that are not the extreme parts, the science fiction come to life parts that a lot of the media focuses on. So we're going to be talking about the tool part. And yes, there are ethical concerns, but we want to understand how the tool works so it's not just a science is messing with what they shouldn't mess with, and understand the actual biology of it, okay? So as we learn about the processes and get involved in these, we're going to try not to fall into the new is scary, new is dangerous trap. We will highlight ethical issues as we go along. We've talked a lot about ethics and genes and genetic testing as we've moved through. Recognize that all the things that we've been talking about in terms of gene therapies are tools. How we use them matters, and absolutely how we use them, particularly in humans, is important to think of as a society. So we're not going to gloss over that too heavily. We will be talking about this in detail as we move towards the CRISPR-Cas. But first, let's talk about the actual creation and building of the tools that create biotech. All right, so first, let's discuss a little bit about how GMOs themselves are constructed. This is the basics, and then we're going to go into exactly how this works, okay? We've talked already a little bit about this GMO, and this is how insulin is created as well. You have a host cell. You have this cell containing the gene of interest, okay? Let's say it's creation of insulin. You make a recombinant DNA and plasmid that goes into a specific cell. That cell multiplies and produces the protein, and maybe you keep it at the bacteria, and you harvest that protein to create, say, blood clot therapy or insulin. Or you may insert that gene using some of the techniques we're going to talk about that into another organism. We're going to be talking about, in particular, how do you create first the recombinant plasmid, and then we're going to be talking about after that how you insert it into another organism. But in particular, creating the recombinant plasmid, that's one of the main things we're focusing on. All right, so how do we actually cut and join DNA fragments, also known as using restriction enzymes, okay? So how do we cut them in a way that we can control, right? So restriction enzymes recognize and cut DNA at specific nucleotide sequences called recognition sequences. So you learned about restriction enzymes in some small part in Bio 1140. As we move deeper into the recombinance concept, we're going to talk about them more thoroughly. So the longer the recognition sequence, the less often it's found in the genome, the less frequently the enzyme will cut the DNA. Okay. So bacteria add methyl groups to the recognition sequence of its own DNA to make sure it doesn't cut up itself, because this is a bacterial enzyme. So does anyone remember why bacteria themselves have this tool, the restriction enzymes, to chop up DNA? What's the purpose here? We talked about it briefly, but in this class, but reach back into your memory. What are restriction enzymes? How do they actually work? Anyone remember? Why do bacteria have them? So it's not for errors, actually, not its own errors. And look at the last part. Bacteria are adding methyl groups to the recognition sequence of its own DNA. They're keeping it safe so that it doesn't digest itself. It doesn't want to fix its own errors. What kind of DNA could get into bacteria that it wouldn't want? Not to build resistance to anything. Viral DNA. Everybody's got it. Okay. So, viruses. Bacteriophages in general will inject their DNA into bacteria. Now, our body has a lot of ways of fighting off the virus after it's in our cells, but we are made of many cells. Bacteria, they're single-celled. In order to protect itself, any unrecognized DNA will be sliced up as much as possible in order to prevent it from producing the proper proteins to propagate the viruses to help the bacteria survive bacteriophages. All right, so this is more Amoeba Sisters, but I think they are very helpful in describing things, okay? So this says, hey guys, great costume party. So he's the restriction enzyme because he chops up DNA, okay? So we'll watch a very brief video here. Hold on, let me pull this over actually.
A common technique in genetic engineering is to insert a new gene into a loop of bacterial DNA.
So you may not be able to hear this very well.
The molecular tool used to cut DNA is a restriction enzyme such as EcoR1.
Okay, let me see if I can put the captions on here. All right, here we go.
A common technique in genetic engineering is to insert a new gene into a loop of bacterial DNA called a plasmid. The molecular tool used to cut DNA is a restriction enzyme such as EcoR1. The enzyme has a precise shape that allows it to run along the groove of the double helix, scanning, in the case of EcoR1, for the base letter sequence GAATTC. The enzyme then cuts the plasmid at this specific point, allowing a new piece of DNA to be inserted. When it cuts, EcoR1 leaves a sticky end. This helps the new gene to attach. The joins are then stitched together by another enzyme called DNA ligase. The genetically engineered bacteria is grown in a culture medium. Very quickly, large numbers of the bacteria can be produced, each with a copy of the inserted gene. The bacteria duly manufacture whatever protein the gene codes for, and so the desired product is made.
All right. Can you guys hear that when I play them? I don't know if it picks up my when I'm not wearing my mic. I wonder if it actually comes up. Okay. Oh, look at that! You could already hear it. Good. Oh, perfect. The one that was coming up next was gene editing with CRISPR-Cas9. We'll talk about that later. All right, but great. So we have so this is just a short information again. About how specific restriction enzymes are used to chop up and then add new chunks. Okay? There we go. So some restriction enzymes, such as HIND3, make staggered cuts in DNA. So does EcoR1. Again, EcoR1 will and HIND3 make staggered cuts in the DNA. Sticky ends, okay? To make sticky ends, you need to have this AG, so you have this AAGCTT. Note the back and forth here, okay? If you have a back and forth, it cuts at two segments in different parts. When it cuts, it leaves the sticky ends. Okay, some restriction enzymes cut both strands across, producing blunt ends. In terms of how this is necessary for DNA, for biotech, a lot of them are actually using the sticky ends because that helps to attach a new segment on after it's pulled apart. Okay. So here we have, okay, a plasmid. This plasmid has this EcoR1 site, okay? And you have this target gene that also has these EcoR1 sites on either end of it, okay? So you flank it, the target gene, the one that you want to insert with the EcoR1, and you have on the plasmid a plasmid that you know has the EcoR1 site as well. When you insert the, when you put this plasmid, when you put the enzyme into this plasmid, you will cut the site and you will break off the ends of the target gene. So now you have these sticky ends flopping around like it did in the video. And these are going to stick in here. Now, sometimes, instead of sticking in, this plasmid will close. And so I'm going to talk about the ways that they figure out if they've been successful with it, because not every single bacterial, not every single plasmid will pull up this. And then once the plasmid is there, you can have the bacteria take it up, and not every single bacteria will take that up. So there's different techniques to find out whether or not the plasmid was successful, and other techniques to figure out whether or not the bacteria took up the plasmid. So you have two sites to check, basically. Okay, so now you have the target gene. It sticks in the plasmid. It fits in this nice sticky part on the sticky part. Now, note that we don't want this to be stuck in upside down. And so because of that, there's a specific way to figure that out too. Okay, so we're going to talk a little bit about that. All right. Again, this is from your book. You cleave it with EcoR1, CTTAA sticking out, AATT sticking out. And then these can stick directly together. And once they're stuck together with the hydrogen bonds, our body can detect, well, bacterial bodies can detect that those double breaks and go fix it with DNA ligase. Okay. All right. So when we chop up a plasmid, we want to be able to, one of the ways, we've talked a little bit about mapping. One of the ways that we can find out how many sites are in the chromosome, or whether or not you've digested the chromosome properly using the restriction enzymes, is to look at the fragments after chopping them up. Okay, so before we are creating the before we're creating the actual product, chopping up the plasmid and knowing is this how many sites are on the plasmid, and also seeing if you've fully digested the plasmid, you can visualize the DNA fragments that come from it. Okay, so viewing DNA fragments. So again, this is another piece that was done in lab. We use, so this was Bio 1140 lab, chopping up plasmids and DNA bits, and then viewing them through gel electrophoresis. These fluorescent dyes. Basic idea is that DNA samples containing fragments of different sizes are placed in wells in an agarose gel. You put the current through it, and you get these fragments. Okay, large fragments travel slower, small ones travel faster, and you have this banding. Okay, so we can look here. Oh, it's doing this again to me. Okay, hold on. Go back. So you've already learned a little bit about this. I'm gonna refresh everyone's memory. Oh, this one I just had. Here you go. Okay. All right. Okay, some of these that I link, you know, if they use these things. So here we go. All right. Again, you have a small plastic tube. You want to find DNA strands of different lengths. You want to find out what those links are. Okay. Oh, is this gonna do it? It may not work. Oh, it's a little angry at me. So. Okay. I'm not gonna stress out about whether or not I can make this work. Let's turn that off. Okay. Our focus is not really to learn about whether or not how gel electrophoresis works. We don't have to actually do that. We don't have that part of in our lab. So instead, the concept here though is that this can be used in, you may have seen these in DNA processing and such. Each individual person, their strands of DNA, when they're treated with these, have different lengths of things. So you have these standard sizes. So you know this is this long, this is this long, this is this long. If you find one that's here that matches here, it's about that long. Vague concept here, okay? Again, different people with different sequences of DNA will have different lengths after being treated with a restriction enzyme. And so you can match up the lengths of those to the lengths there. You don't have to have any sequencing at all. You can do the banding here just to understand the different lengths as you treat them with restriction enzymes, okay? All right. So again, after you have this gel, then you lay the film on the gel, you keep it in the dark, you develop the film. Okay, so now you don't have to hold the gel, you can actually just hold up basically a picture of the gel. So now you have the positions of the bands. All right. But so I taught you guys this already, but the purpose of the restriction enzyme is to help the bacteria. What? Okay. Fania says D. Beatrice says D. Okay. And that's the thing we just said, okay? Defense against virus attack, cutting up foreign DNA. Again, this is something where we don't have the restriction enzymes in our body. We have a ton of DNA and no defense against that. We don't have the same things that the bacteria has. But we also are multicellular, and that's not how we kill off viruses. We send our white blood cells to go and attack and destroy the virus. But obviously DNA do not have white blood cells. This is their form of a white blood cell, okay? To destroy them. All right, so let's take these random restriction enzymes, okay? 5' CTG CAG, okay? And then we have restriction B, recognition sequence GCG C. So what can you infer about the enzyme based on how it's recognizing and cutting? So it's a little tricky, I know. All right. I'm gonna give you guys a hint here. The deal is that the sticky ends, you can't actually, neither one of these is going to create sticky ends. You can't tell if it's going to create blunt ends either, okay? The deal here, though, is that the sticky ends would have a repeated and then a flipped and repeated to make the sticky, because it would cut and then cut, okay, on the opposite sides, okay? But that's not happening in either of these, okay? This GC would not cut it properly, okay? It's going to actually be this one. So you can't actually tell whether it's blunt or sticky. I can tell you that neither of them are sticky. So C and D are wrong, okay? Neither of them are sticky. Again, it would because this here, it would not stick onto here, okay? That's how the sticky is created, we'll walk through the sticky part in a minute. But the idea is that enzyme A, because it recognizes a longer sequence, it will cut the genome into fewer pieces than enzyme B. Again, one of the ways that you can figure out whether or not your restriction enzyme has been useful, has been actually successful, is the way that it cuts the genome, and you can count up, okay? But, all right, let's move on, and then we'll come back to restriction enzymes a little bit. So now we also have to figure out how to locate DNA fragments, okay? So DNA containing a certain gene of interest, you want to extract it from human cells and cut it into fragments, and you want to figure out, did I find the right fragment, the right gene size? Okay, so you know the gene length. So you chop it up and you figure out, so you separate them by size. Each band consists of many copies of a particular DNA fragment, okay? So then you blot it, and you, the key here is that you have a radioactively labeled probe, okay, for a specific gene. So this is going to target a spot on the gene and say, which of these fragments is the fragment that I want, that has the gene on it. So figure, okay, the fragment that I want in this specific, let's say human DNA, you're looking for the fragment that contains the insulin gene, okay? So you have a probe that is looking for the specific insulin one. It highlights this. Okay, so now we know that the insulin one is on the one that's this long. And then with more information, we can actually figure out what the actual sequence is of that specific fragment. Okay, so now you've targeted your fragment. You know which fragment you're trying to find and amplify, and you can amplify that fragment. All right. We're not doing much with DNA gel electrophoresis, so I'm going to skip this one mostly. We already talked about larger ones go one way, smaller DNA go another, okay? Then identifying the fragments past that. Okay. So let's talk first about the cloning of genes themselves, okay? Then we'll talk about applying and then amplifying. I don't think we're going to get to these second two. Well, we're going to talk a lot about cloning, and then that'll be the rest of today, okay? All right. So gene cloning. You want to amplify a specific piece of DNA via a bacterial cell. So you need a cloning vector, okay? It's a molecule attached with a foreign DNA fragment to be introduced into a cell. All right. So you're creating these plasmids. So this is part of the big piece, and we talked about this a little bit before, is identifying now, being able to have these, you have these like, these individual created plasmids, okay? You have a cloning vector, contains an origin of replication recognized in the host cell. So it is replicated along with the DNA that it carries, okay? So this is going to be the part where it's going to attach along with the gene that's there, okay? So then you have a single cleavage site for one or more restriction enzyme used. Again, usually they have multiple ones, but you only treat it with one based on the specific cloning vector that you're using. And then selectable markers, okay? Traits that enable cells containing the vector to be selected or identified. Let's walk through these, okay? The concept here, okay? You have multiple cloning site. You have all of these possible ones to use, okay? So this is just the base one that everyone can use. And you have all these different spaces in here that can be cut by any one of these, okay? You highlight whatever gene that you're interested in. Again, we don't have all these genes, and we don't have all the restriction enzymes, but the concept here is that you highlight whichever one is surrounded by one of these, and then you only add that restriction enzyme, and it only occurs here on the plasmid, cuts in one space. Again, these are generally sticky-ended, and when they cut in one space, it will open it up to only accept the gene of interest. And you'll note here, this is the lax Z gene, okay? And I'm going to talk about why we need this part in a minute. But so now you have this, and you want to put it into a bacteria. You want to and then you want to take the bacteria and have the bacteria infect, let's say, another organism. And so this is a resistance gene. So if you treat whatever, if you treat something with this ampicillin, then you'll know which ones have pulled it up and can use the resistance gene. And you'll know that they have the inserted DNA. Okay? So the goal here is that the restriction sites are not present anywhere else in the plasmid. When you cut the plasmid, you're only cutting in one site, and that's specific to the gene you want to insert. It does contain multiple cloning sites and multiple cleaving areas. You just choose the one that will correspond to the inserted the DNA that you want to create the vector for. Okay. All right. So let's say you have the insulin gene. Okay. The insulin gene. So now you have all the restriction enzymes here. You have the target gene, and this has a certain restriction site on one side and a certain restriction site on the other side. Okay. You digest both of these with that one restriction enzyme. This creates sticky ends, and that's joined with the DNA ligase. Now you have a recombinant plasmid. Then you take the products of this recombinant plasmid, or what you think are the recombinant plasmids. Not every plasmid is going to be recombinant after you create it, but the concept here is that you want to figure out, you'll figure that out in a minute, okay? But first you take these what you think are recombinant plasmids and put them in with bacteria. In a heat shock, which again was done in Bio 1140, you force the bacteria to take up a plasmid. As they take up that plasmid, some of them can then start using the insulin gene to build insulin. Okay? To figure out which one, you also have this antibiotic resistance gene. Okay? The antibiotic resistance gene can be used along with the insulin. Now you can't see the insulin immediately, but you can see the antibiotic resistance immediately because you place the bacteria on an antibiotic plate. Only the ones that contain this gene survive, and those are the ones that have the recombinant plasmid. Then we have the colonies grown in a large culture, and you induce them to express the gene. Now you have the mRNA translated into protein, like the insulin, and that's how you make insulin factories. This is a little bit more in detail of what we went through in the case study, but you can see how this would work reasonably well. Now, the next step we're going to look at is this first piece. One of the things is that, so we have this experimental plasmid. It can be used for multiple types of this, okay? But you want to be able to be sure it's recombinant, okay? And I showed you this picture before. This is the lax Z gene. Okay, so the next piece here is to figure out whether or not you've been successful in creating a, not just one that has not just one that has the plasmid, but did it pick up a recombinant plasmid, or does some of the plasmids fail at picking up the gene of interest, okay, target gene. All right. So again, we have the plasmid vector cut with a restriction enzyme. DNA to be cloned is cut with the same restriction enzyme. They stick together at the right spot. Okay. Now you want to collect not just, so there's 3 types of cells now that can result from this. You have a host cell with its chromosome and no plasmid. Well, that you don't want. This one will be killed off by the antibacterial one because, again, it needs that antibacterial that's on the plasmid to survive. But these two both have the antibacterial gene. You want to only select this one. So now you have a second one called the color indicator, okay? That lacks Z gene, okay? So again, sometimes when you're creating the recombinant plasmid, it doesn't become recombinant. It won't pick it up. So you have this lax Z gene that have, again, these sites, the sites that can be cut, okay? So only cells with the resistant plasmid grow selective medium, and now you have to find the recombinant ones, okay? Plasmids that have an insert that disrupt the lax Z gene, note that it would insert here and break up the lax Z gene, and instead of reading a full lax Z gene, you're going to have it read an incomplete, wrong lax Z gene. So now it can't metabolize a target compound that produces the blue color. Okay, lax Z would be able to metabolize that target compound. So blue colonies are the ones that are bad, right? This is the blue-white ones, okay? White colonies have recombinant plasmids. So now you've identified both. They've taken up the plasmid. We know this because they survive on the antibacterial medium. And they have recombinant plasmids, which is identified by the breaking up of that lax Z gene that surrounds the recombinant sites, okay? So this is kind of the concept I want you guys to be able to understand. All right. So again, here you go, sticky ends, etc. This is just another explanation. Cutting the lax Z gene prevents the eventual production of the enzyme that would be able to break down that blue color. That would be able to create that blue color. So this would tell you that you had a recombinant one. You have to put them in the bacteria first. Once you have the bacteria, you can then induce the bacteria itself to infect a germline cell of another organism, like a plant. You take this plant cell and you infect it with that. Again, do the same concept, put it onto, say, a pesticide. You have a pesticide resistance gene in there, and only the ones that have the pesticide resistance gene and your gene of interest will grow. And then you know that you have a recombinant and transgenic plant. Okay, so the same concept, except now you'd put the bacteria into the plant. All right. So this is the whole process, but you guys can go through that again, okay? The next thing you want to do is figure out if your gene of interest is inserted correctly into the plasmid. Obviously, we already talked about the plasmid closing back up, and we wanted to have done it forward, but what if it goes in backwards? You want to now instead, you need to now do a restriction digest, okay? So the key here is that if it inserts properly, then you'll see, you'll be able to identify whether it's gone in forward, which will create this pattern, or reverse, which creates this pattern, okay? And so it's where the resistance ones have gone in reference, okay? So when you break it down and you measure them, you can see where they, whether it's forward or reverse, okay? So again, you want to make sure that it's been digested properly and that it's been inserted properly, okay? So then you will take this and when it's broken up by only the HIND3 or the BAMH1 or both of them, it's going to have a different setup, okay? the sum of the fragments equals the size of the unfragmented DNA molecule. This is one way to figure out the length of your DNA molecule, but think of this as sort of like another way of making a map. Okay, so the map shows the order of the fragments and distance between the restriction sites. Okay, when you have HIND3 and BAMH1, okay, BAMH1 is going to cut at 1.8 and 3.1, okay, and HIND3 is only going to cut at 2.8. But then you're going to expect one that's 1.8 long, and 1 long, and 0.3 long, and 0.9 long. If you see either one of these, only one of the restriction has done, or if you don't see either of those, then you'll see that the neither one of those has properly digested it. Okay. All right. So, well, let's walk through one of these, and then that'll be the end here, okay? So we want to find out if we have been successful we want to know what's going to happen if we have succeeded at cutting it both with EcoR1 and XBA1, okay? So, can someone tell me what the length of one of the bands is going to be? What's the length of the first band you're going to see? What's the length of the first band? You just go down the line and figure out. If you know that it goes at 10 and 28, this one goes at 6, 18, and 38. Oop, move too soon. Okay, we have 6 first, and then what's this one? 6 to 10. You're going to see a 4. 10 to 18. You're going to see an 8. 18 to 28. You're going to see a 10. 28 to 38. You're going to see a 10, and 38 to 42. Okay. So let's look at the answer, and we can see. Okay. So you see that's different whether it's cut by 1 or both. So if it's cut by 1, you're going to see this pattern: 18, 14, 10. Okay, because obviously that makes sense. 18. And then there's sorry. This is the 18 one. Okay, this is the 10 one, and this is the 14 one. Okay. If it's cut by the XBA1, you have one that's 20 kilobytes long, okay? That's this one. You have one that's 12 kilobytes long. That's this one, okay? One that's 6, well, that's the first part, and you have one that's 4, that's the last part, okay? Again, we see this 10, 8, 6, 4 like we expected instead, okay? Because there's actually multiples in one of these, right? Because we had 2 8s, or no, 2 10s. We had 2 10s. So this 20 kilobyte was cut into 2 10s. And you might not see that many bands, but that's the concept, okay? So I want you guys to be able to recognize how to do that, okay? All right. So if I gave you guys something like this on the final, do you see how you would do it, or do you want me to walk through it one more time? Either way, we have time. All right, one more time. I can do it. Okay. Let's start with one of them. EcoR1, if you cut, so you know the length of this band. You know how you know the length of the band? What you're going to do is you're going to run it with no you're going to run it with only, let's say, a site that you know is going to create a blunt end, and it's going to, you're going to be able to get the length of it, okay? So we run it with a site to linearize that plasmid, okay? We know now it's a 42 kilobyte plasmid. Okay. So you have that information. So now you want to find out, were you successful at cutting it with the EcoRye? Were you successful indeed in opening it, okay? If a 42 one is cut, you look at this fragment, again, only with the EcoRye, you have a 10 fragment, okay? See that 10 fragment? You will expect something to show up at this 10 size. Where this is lower down, it travels down faster, so you're going to see it at the 10 kilobyte, okay? This is your size marker. 10. Then you have 10 to 28, that's 18. There's your 18 right there. 28 to 42, that's 14. That's 14 right there. Okay? That is EcoR1. Now if you cut it only with the XBA one, 0 to 6, there's a 6 length. 6 to 18, there's a 12 length. Okay, that's the 12. 18 to 38, there's a 20 length. There's the 20. And then 28 to 38, there's a 10 length. Oh no, sorry, not 28. Forget 28. 28 is not there. 38 to 42, that's the final one. Okay, that's a 4. Okay, so you have the 6, the 12, the 20, the 4. All right, hopefully that's reasonably clear. Now we look at this one. It only has 4, but that's because some of them are the same size. So let's walk through that, okay? It's going to have more than 4 fragments, but you're only going to see 4 bands because there's some multiples. 0 to 6, there's a 6 right there. 6 to 10, there's a 4 right there. So that's where the 6 and 4 are coming from. 10 to 18, there's an 8. Now note, 18 to 28, again, cut here and cut here. 18 to 28 is 10. 28 to 38 is 10. 38 to 42 is 4. So there's 2 4s, one from the 6 to 10, one from the 38 to 42. Those are both in the 4 slot. You're not going to be able to tell how many fragments are there because, for one thing, you're going to have multiples of these, so it doesn't matter that there's like 2 instead of but there's really 200 because you're putting a lot of these in there. Okay? And then you have 2 10s, the 18 to 28 and the 28 to 38. Okay? So that's where that 10s come from. EcoR1 itself has a 10 has a 10 length as well, okay? Because it has a 0 to 10 fragment. Right? So you're going to see the 10 in both of these, but they're going to be actually different fragments, 18 to 28 and 28 to 38, instead of 0 to 10, because 0 to 10 is chopped up by the other XBA one. The linear restriction map is to gauge distance between the cuts. Don't worry about how many cuts there are. Well, do worry about how many cuts, but the idea is that you're not trying to count the fragments. You're trying to figure out how many bands you expect to see and what patterns you're going to expect to see if you are cutting with one versus the other. Okay? All right. Now that I walk through it bit by bit, everyone else understand this? All right, good. Great. Okay. We are going to walk through the homework. As I mentioned, I will show you the one that I gave everyone credit for because there's an error. So we'll walk through that in a minute. But let's just go for like 12 minutes from now so that we have a good, nice round number 3:35. Okay, 3:35. Give you a full just over 10 minutes to reset, get yourself something to drink, get your Zoom set up. Okay, put the Zoom like I normally do into here. Oop, that's not what I must do. Okay, give me a minute. I'll pull it up. I'll pull it up. Okay, pull up my Zoom and set it up. All right, here's your Zoom meeting. Meet here at 3:35. There you go. Okay. I'll see you guys then when I end this for now, and I'll post this and the PowerPoint later this evening. Your final homework and your final case study are posted, so you can work on those at any time. Okay, and next week we will have one review quiz on Blackboard. We have one extra case study, so everything all balances out. Okay, see you guys there.